First of all, thank you for inviting me to this nice conference. I mean, as Joanna said, that I'm working as a postdoctoral researcher in the lab of Professor Hagn, and in our lab, we work on structural biology of membrane proteins. And today I will talk about our recent paper on structural basis of apoptosis induction by mitochondrial VDAC. And actually, this work is already in bioRxiv if you are interested, and also it's at the moment under revision in Nature Communications. As you may already, as you may already know, VDAC is found in the mitochondrial outer membrane, and its main function is transport of small molecules across the outer membrane, like ATP, ADP, or metabolites, or small ions. So this protein is comprised of 19 beta strands, making this nice beta barrel channel, and also it has an N-terminal alpha helical domain, which is residing inside the pore in the canonical conformation. So this protein, in addition to its main function as a transport of molecules, it has been also shown to act as a key player in the apoptosis mechanism. So during apoptosis, upon an apoptosis signal, mitochondrial outer membrane gets permeabilized, and the cytochrome C release from the intermembrane space actually leads to cell death. However, how VDAC is really mechanistically regulating this process is not really clear. In the literature, it has been clearly shown that the VDAC gets oligomerized upon different variety of apoptotic stimuli, and also it has been shown that this protein can interact with pro and anti-apoptotic proteins, and this N-terminal helix is actually very important in this case. And even like for the oligomerization of this protein, actually, it has been proposed that the oligomerization might be directly having a role in the formation of this pore formation in the outer membrane. However, until now, we don't have any structural proof for such a confirmation. And also recently, actually, in this year, an AFM study has been shown that VDAC VDAC is oligomerized in a lipid state. However, unlike in this proposed state, in between these oligomers were actually filled with lipid molecules. So this actually proposed mechanism can be questioned actually with this paper. And in our story actually begins with, so what we think that since this N-terminal domain of VDAC is binding with the pro-apoptotic or anti-apoptotic proteins, it needs to be exposed to the outside of the pore. However, such a structure until now has not been shown. And what we think is this oligomerization of VDAC in the membrane might be a trigger for such an exposure for the N-terminal domain of VDAC. And in our story, first we wanted to probe the helix exposure to outside of the molecule barrel. So we used a single cysteine mutant at the very end terminus of the protein, which is normally inside the beta barrel. And by adding of a maleimide molecule, which is 40 kilodalton in size, so it should not access when it's inside the pore. However, when it's exposed in the solution, then this molecule can modify this N-terminal region easily. And actually, fortunately, we were able to, in the end of our screen, we were able to find a condition that a negatively charged cholate as the detergent actually significantly increased this exposure in the exposure of the N-terminal helix to the outside of the pore. And as we predicted, this event was actually quite coupled to the oligomerization of the VDAC as monitored with this chemical cross-linking, amino-specific chemical cross-linking experiment. under same conditions with this negatively charged cholate detergent. And then as a control, we also used another mutant on top of this T60 mutation. So there is in redac, there is a glutamic acid residue at this membrane-facing side of the channel, and mutation of this residue to a valine, so more hydrophobic residue, actually increases the stability of the beta-barrel channel, and then this keeps and this actually decreases the oligomerization tendency of the protein, as you can clearly see here. And as confirmed with this experiment, actually, what we also saw, observe is when the oligomerization is reduced, we also see a reduction in the exposure of the N-terminus of VDAC also in this mutant. And of course, detergents are not the native environment for this protein. So we wanted to do the very same experiment also in liposomes. So here we used POPC as a lipid, and we observed actually very similar result in the presence of E73V mutation. We observed the modification in this of the N-terminus is slightly reduced. However, this time the effects of this exposure and also the oligomerization was not as pronounced as in the detergent case, but we see actually similar trends for this. And again, to mimic the cholate, actually to come more close to the cholate state, we also added a negatively charged lipid, POPG here, in our liposomes, and we found that in this case, actually, the exposure of the N-terminal helix is increased in this case, actually confirming our model that oligomerization of VDAC in the membrane is inducing the exposure of its N-terminus, and actually negative charges on the lipid surface seems to have a stabilization effect in this conformational change. Then we wondered actually with a structural tool if we can really monitor this exposure of the helix or not. We wanted to use our nanodisk technology as a tool for conformational trapping of VDAC in a helix exposed state. And for this, what we did, we used like different sized MSP nanodisks, and checked the helix exposure again with this PM40 modification assay. And what we found here is this larger nanodisk, which give like enough space to VDAC for its like canonical helix inside conformation. And the smaller nanodisks generated a more confined environment, and then the helix goes out of the barrel. And actually, we think that this smaller nanodisk is actually mimicking the oligomerization state of this protein for our, and we can use this for our analysis for our experiments. And first of all, we wanted to check this with NMR with a specifically methionine, specifically methionine labeled protein. And we have also additional methionine mutation in the very N-terminus of the protein, and also these two native methionines on the wall. What we observe with the larger nanodisc, actually this protein is present in two conformations. So we see for this 155 and V3M methionines, we see two different conformations. And addition of BCLXL, which is an anti-apoptotic protein, as a partner of as a binding partner of the N-terminal part of VDAC, we observe only slight changes in one of these conformations. One of these conformations showing that this VDAC is found as in a conformational equilibrium in larger nanodiscs. However, when we use the smaller nanodisk, so then we just see only one conformation in this case, and here the peaks are a bit broadened because of the instability. Because of the interaction between the MSP belt and also the membrane protein. But what we see basically is in this time, addition of BCL-XL showed actually a direct, so the line broadening in this N-terminal mutant. And as I said, it's just one conformation. So it looks like our biochemical experiments actually confirmed by our NMR experiments. And as a next step, we wanted to see these particles also using cryo-EM. And in cryo-EM, when we use this large nanodiscs, VDAC is, we found VDAC either as in a monomeric state or in a dimeric state in these nanodiscs. And interestingly, in both states, we find an electron density inside the barrel, which can nicely, which you can nicely see in this figure. However, when we used smaller nanodiscs, if we could not detect any electron density for the N-terminal helix, which suggests and also confirms our biochemical and NMR experiments for the exposure of N-terminal helix. Okay, now actually we for the first time showed the helix exposure state is actually exists with a structural method, and we wanted to check if this outstate, the helix outstate can bind to its interaction partner, be anti-apoptotic BCL-XL. And here you can see the NMR spectrum of BCL-XL, and addition of the smaller nanodiscs on top of this on this sample actually induce chemical shift perturbations at specific residues in this protein. However, in the larger nanodiscs, we do not see this effect as much as in the smaller nanodiscs. And when we map this chemical shift perturbations on the structure of BCL-XL, what we observe is actually these chemical shift perturbations are clustered around the BH3 binding side of BAC. So BAC is a pro-apoptotic protein. It makes pores on the mitochondrial outer membrane upon apoptotic signal. And actually, to check this, we did one more control experiment. So we repeated this experiment, but in the presence of a BH3 helix of BAC. And what we see here, as in the larger nanodisc case, we did not see any change supporting that, okay, this BH3 binding side of BCL-XL is competed by with the N-terminal domain of VDAC and also the BH3 helix of BAC. Then we would like to, we wanted to get a high resolution structure of this complex. However, the affinity of this helix to the BCL-XL was in low micromolar range. I mean, it's still a good binding affinity. However, for the crystallization experiments, we utilized the fusion construct that's made where the VDAC N-terminal domain is attached to the BCL-XL C-terminal part. And this actually yielded a very nice crystal structure at high resolution, showing this complex and also confirming our NMR experiments, NMR chemical shift experiments. Then afterwards, to see if this complex is really physiologically relevant or not, we did a short molecular dynamic simulations to see in a lipid membrane environment setup. So what we see here, so this is our structure, and the beta barrel is in the lipid membrane. And as you can see in the complex formation, the C-terminal tail of BCL-XL is close to membrane because normally that has a transmembrane segment, and this looks like feasible for under physiological conditions as well. And of course, afterwards, we wanted to also check the via functional assays. So here you see a pore-forming assay, which is used widely in this apoptotic proteins. And BAC as a pro-apoptotic protein actually makes forms at high concentrations, and it gets auto-activated. And you can see this nice pore formation as the increase in fluorescence. And addition of BCL-XL, which is an anti-apoptotic protein, which inhibits BAC under normal conditions, this pore formation is actually attenuated. And interestingly, and as we expected, addition of N-terminal peptide of VDAC, actually we observe this inhibition of inhibition in a concentration dependent manner in this functional assay. And of course, this effect can be also through the Bach directly. So in a control experiment we checked if Bach in the lower concentration needs to be activated with activator proteins such as CBIT. And we checked if VDAC1 N-terminal domain in excess amount can activate pore formation or not. And it looks like it cannot. And this actually shows already that VDAC looks like to directly affect BCL-XL inhibition, so inhibit the BCL-XL inhibition of Bach pore formation. Actually, with that, I come to our final model. So VDAC actually, it looks like, unlike it's said in the literature from our model, VDAC does not have a direct role in the permeabilization of the outer mitochondrial membrane. It's rather act as a sensitizer, a BH3 protein to induce apoptosis. So under normal conditions, the protein is in a monomeric state. Monomeric state where the helix is inside. However, in the when upon like apoptotic stimuli, mitochondrial damage, and upregulation, so VDAC gets oligomerized, which we don't know how this is really regulated, but this requires further investigation. The N-terminal helix of VDAC can get exposed to the cytosolic side of the mitochondria, and this competes with Bach for BCL-XL binding, and in the end, Bach stays free when the BCL-XL is inhibited and the pore formation occurs. So here, actually, VDAC is, as a sensitizer protein, is inducing apoptosis upon specific signal. And with this, I would like to thank you for your attention, and also our lab members and also all the contributors, our cryo-EM collaborators, and also for the crystallization, our collaborators in Helmholtz. And